Hey everyone! Today we're going to continue on with chapter 3 and we're going to focus on continuing learning about the cell. So, in the last lesson, we introduced the cell and we talked about some things that cells have in common. So, before we continue on, we're going to do a little bit of a review. In the last lesson, we talked about what are cells. Well, cells are the structural units of all living things. In other words, they are the building blocks of life. There are trillions of cells in the human body, and there are over 200 different cell types. These cells can vary based on size, shape, and function. Now, since no one type of cell is exactly like the others, we do know that cells have some basic parts or certain functions that they have in common. So, what we started discussing in the last lesson was what does a generalized cell looks like? So, in general, cells have three main regions or parts. The first part is the nucleus, the second is the plasma membrane, and the third is the cytoplasm. We talked about the nucleus and the plasma membrane in the last lesson, and this lesson we're going to focus on cytoplasm. Before we get to the cytoplasm, let's do a quick review of the nucleus and plasma membrane. In terms of the nucleus, we talked about how it was the control center of the cell. This is where you're going to find genetic material, known as DNA, that is needed to build proteins and for mitosis, in other words, cell reproduction. There are three parts of the nucleus. You have the nuclear envelope, which is going to surround the nucleus and allow things inside the nucleus and certain things outside the nucleus. You have that dense middle region known as the nucleolus, where ribosomes are going to be created. And then you have chromatin, which is scattered throughout the nucleus, and they condense to form chromosomes during DNA replication, or basically mitosis as well. So here we have the nucleus. You can see that we have the nuclear envelope with the chromatin scattered throughout the nucleus, and then the dense middle where the nucleolus is formed. We also then moved on to the plasma membrane. The plasma membrane is a transparent barrier. So it's a barrier for the cell contents located within the cell. So it contains those contents and it keeps them away from or separate from the surrounding environment, such as other cells outside of it or even the extracellular fluid. Here we brought up the general picture of a plasma membrane. Remember, the plasma membrane is made up of phospholipids. It's a phospholipid bilayer, so there are two layers to it. You have your hydrophilic heads, and then your hydrophobic tails, and they're lined up tail to tail. Also remember that the plasma membrane has proteins located throughout in order to help facilitate for diffusion, helps facilitate active transport and moving things across the membrane. We also have cholesterol, and sugar can be found in the plasma membrane as well. So, your cytoplasm is what we're going to be talking about and focusing on today. This is where your notes are going to pick up. So, what is the cytoplasm? It is the cellular material outside of the nucleus, but inside that plasma membrane. This is going to be the site of most cellular activities. The activities can include, but are not limited to, digestion, metabolism. This is also where you're going to see them excrete waste, break down products that they do not need, but also start building products that they do need, such as proteins. You're going to see movement within the cytoplasm, different organelles moving around, sending vesicles everywhere. So the cytoplasm does a lot of the necessary life functions that we learned about in the first chapter. The cytoplasm includes three main parts: your cytosol, inclusions, and organelles. So let's define those three major components. The first, cytosol. Oftentimes, people interchange cytoplasm and cytosol, But cytoplasm is the overall major region outside of that nucleus. Cytosol is just the fluid. This is the fluid that helps suspend elements. This usually contains nutrients and electrolytes that the cells are going to need. Then you have inclusions. These are chemical substances that are free floating around in that cytosol, such as those nutrients or cell products or even waste that needs to be gotten rid of. They float in the cytosol. And the last, that's going to be our primary focus, are organelles. Organelles are usually covered in a regular general biology class. Organelles are defined as the metabolic machinery of the cell, and they perform functions for the cell. Organelles are membrane bound, and the reason they're membrane bound, especially in a eukaryotic cell such as us humans, is they help compartmentalize their functions. So their functions retain and stay at those organelles. Here is your book picture of the general cell. Again, we talked about the nucleus and we talked about the plasma membrane, but now we're going to talk about all of the stuff here. So we're going to talk about the smooth ER. We're going to talk about lysosomes and mitochondria and centrioles. We're even going to focus on things like the Golgi apparatus, rough ER, and ribosomes. So let's start talking about these organelles. If I ask any kid what do they remember from biology and science, most people can tell me mitochondria's function, which is the powerhouse of the cell. Mitochondria are structured by having a double membrane that have little curves or folds in it called cristae, and these are located on your inner membrane. So if I was going to go back and look at a picture of mitochondria, here, this inner folds here, these are the cristae that they're talking about. These carry out functions where oxygen is used to break down food and make ATP molecules. This is where cellular respiration is going to take place within a cell. So remember, ATP makes energy. It is the site of cellular respiration. The next major organelle that we're going to talk about are ribosomes. Ribosomes are very important. I guarantee you I'll ask you a question on the test. They are made up of proteins and ribosomal RNA. So RNA, let's review what that is. RNA is single-stranded form of DNA. So instead of a double strand like deoxy DNA, it's going to be a single strand. Also, the base units are different. Instead of the base unit thymine, it has uracil. There are different types of RNA, and the one that makes ribosomes is ribosomal RNA. Ribosomes are important because they are the site of protein synthesis in the cell. They are in charge of where translation is going to be, if you remember the terms transcription and translation from biology. Ribosomes can be found in two locations. They can be free-floating within the cytoplasm, not attached to anything, or sometimes ribosomes like to attach themselves to the rough endoplasmic reticulum, also known as the rough ER. The next major organelle we have is the endoplasmic reticulum, which we just abbreviate ER. These are fluid-filled tunnels or canals that carry substances within the cell. They carry substances through something called vesicles, which basically act as tiny cars that carry chemicals, compounds, proteins, lipids from one part of the cell to the other. They are generally continuous. This one are generally continuous with the nuclear membrane. And there are two types, rough ER and smooth ER. So I'm going to go back and show you a picture of it. Here, again, it's continuous with your nuclear membrane, so usually you're going to see it surrounding the nucleus. Here is a picture of your rough ER. You can see the little orange dots. Those are the ribosomes stuck to it. Whereas if you go to the smooth ER over here, it's also still a continuation. It's blue. You're not going to see any orange dots because there's no ribosome stuck to it. Again, you can also see ribosomes free floating all throughout the cell as well. So let's go a little bit more detail about rough ER. Well, we already stated that it is studded with ribosomes. Again, ribosomes are in charge of protein synthesis. So rough ER helps the synthesize proteins. The most important thing that they do is they help transport these proteins that are being made using vesicles, or basically the cars of the cell. And they move these proteins throughout the cell and even all the way to the cell membrane to be released to your body. Again, rough ER is going to be abundant in cells that do a major function of making and exporting proteins. Here is a picture of your rough ER where a protein was made on a ribosome. It goes through the ER tunnel system where it is then put into a vesicle or a little bud, and it is then transported to wherever it needs to go. For example, in this picture, it's saying that it's going to travel to a structure that we're going to talk about in a few slides called the Golgi apparatus. The smooth ER is called smooth ER because it lacks ribosomes. So it does not have a function in protein synthesis, but it does function in something called lipid or steroid synthesis. So remember, lipids usually have to do with fats. The smooth ER also helps detoxify drugs and pesticides that may enter your body. The next one that we're going to look at is we've mentioned the Golgi apparatus. It sometimes receives proteins coming from your ER. They appear as stacks of flattened membranes, and they're associated with tiny vesicles, or those tiny moving, basically cars of the cell taking materials to and from. What happens is it gets packages from the rough ER, and it modifies those packages and those proteins and then decides what to do with them. When it gets these proteins and it modifies them, it can take one of three pathways. It can be a secretory vesicle, means it secretes something that was made. It could keep those proteins and lipids in-house and then move them where they need to be. Or it can create something called lysosomes and send things to lysosomes. Here is a picture of what your Golgi apparatus is going to look like. Again, there are a lot of folds. They usually have to do with vesicles. So let's start back up here at the ER. Here's your ER. It has some proteins in it. What's going to happen is that's going to bud off and come over here to your Golgi apparatus, where it's going to drop off what was created. From here, your Golgi apparatus is going to refine this, it's going to package it up, and then decide what to do. One pathway that it can do is it can take that protein or whatever was created and it can take it over to the plasma membrane and then release it out to the rest of the cells, release it out of the cell to, let's say, a nearby tissue or nearby organ or into the bloodstream. A second path that it can do is it can actually decide to make components that are used in-house. For example, this one is making components that are going to be used on the plasma membrane. So it takes it over to the plasma membrane and it attaches it. So you can use it here. Or, for example, if, let's say, the centrioles needed something, it would take it over inside the cell and you can use it over there. The last pathway is what happens is these let's say proteins, bud off and they get taken to something called a lysosome, where the lysosome is going to break down and digest these substances because they aren't needed. So let's focus on a lysosome now that we mentioned it. Lysosomes are bags, membranous bags, that contain digestive enzymes. These enzymes break down things that are worn out or things that are not needed to be used by the cell, so unneeded cell structures. Lysosomes house phagocytes. Phago means eating. So basically, they ingest these things that are no longer needed and they break them down using the enzymes to get rid of it. And this includes getting rid of bacteria or even cell debris. The next organelle we're going to talk about are called peroxisomes. These are membranous sacs that contain oxidase enzymes. So again, if it ends in ase, it is generally an enzyme. What peroxisomes do is they help detoxify harmful substances like alcohol and formaldehyde. What they do is they break down free radicals that are located within these substances. Free radicals are just highly reactive chemicals. What they do is they take these free radicals and they convert them to hydrogen peroxide or H2O2, hence the name peroxisomes. Hydrogen peroxide then decomposes into water. Peroxisomes are created and replicated by just pinching off and budding off of the ER. Now that we've covered a majority of the organelles, how are all those organelles able to stay within the cell, or what gives the cell the structure that it has? Well, this is where the cytoskeleton comes into play. The cytoskeleton is a network of proteins that go throughout and extend all throughout the cytoplasm. They function in providing the cell with the framework to help determine the cell's shape, support organelles within the cytoplasm, and allows things to be transported along different roads that it provides. So, in other words, think about the framework of a house. There are three different types of elements formed from the cytoskeleton. There are microfilaments, these are the largest. Actually, I have this backwards. This is the smallest. Microtubules are going to be the largest. So please fix that in your notes. So microfilaments are the smallest. Then you have intermediate filaments are in the middle, and then microtubules are the largest. So again, microfilaments, these are going to be things like actin and myosin. These are going to come into play when we talk about the muscular chapter. We have intermediate filaments. These form a purple network surrounding the pink nucleus to help brace it. And then even tubulin subunits, these appear and they actually help with cellular reproduction. Then the last one we're going to talk about are centrioles. Centrioles are generally only found in animal cells. Since we're focusing on anatomy and physiology of animals, especially humans, we have to talk about them. These are rod-shaped bodies and they're made up of microtubules, which is a cytoskeleton. These microtubules form and help direct mitotic spindles during cell division. So what happens is during cell division, we have centrioles that go to opposite ends of the cell. What happens is they release these little spindles that have, and then in the middle, there are usually chromosomes. And these spindles help break the chromosomes apart and move to opposite sides of the cell, where then the cell will split and create two whole new cells. Now that we've talked about all the organelles, the last thing we have to talk about in terms of the cell are cell extensions. Now, outside of the plasma membrane, sometimes you will see some surface extensions. The three that we're going to talk about are cilia, flagella, and microvilli. Cilia, their major thing is to help move materials across a cell surface. They generally move or wave back and forth. For example, in the respiratory system, they help move mucus that have different things trapped into it away from our lungs so we're able to cough it back up. This helps prevent us from getting sick with bacteria. Then you have the flagella. This helps propel cells, and like we said in yesterday's lesson, that is the only flagellated cell in the human body is going to be the sperm. The last one is microvilli. These are tiny finger-like extensions off of the plasma membrane, and these are important to help increase body area for absorption. This is very important, especially for your digestive system, where you're going to be absorbing nutrients. And that's the end of today's lesson. Let me know if you have any questions, and I'll be more than happy to help you. And otherwise, I hope you guys have a great day.